Greetings. You're invited to engage in a video lecture on the glymphatic system, skillfully crafted by a dedicated student from Ivana Tbilisi State University, Faculty of Medicine. I encourage your focused attention as these students have invested significant effort in preparing this presentation. I trust you will not only find it informative but also appreciate the hard work and dedication they have poured into this endeavor. Thank you for your attention, and I hope you thoroughly enjoy the presentation.
The unveiling of the glymphatic system marked a pivotal moment in neuroscience, fundamentally reshaping our comprehension of how the brain rids itself of waste. Unlike conventional pathways, the glymphatic system represents a groundbreaking mechanism for waste clearance. The repercussions of glymphatic dysfunction reverberate across the various neurological conditions spanning from neurodegenerative diseases like Alzheimer's to traumatic brain injuries. Today, we embark on a journey to delve deeper into the complexities of this system, aiming to unravel its mysteries and shed light on its implications.
The glymphatic system is a recently discovered system by the Danish neuroscientist Dr. Michael Negogaard in the year 2013. So what is it? The term glymphatic is a combination of glial cells and lymphatic, highlighting the involvement of glial cells, particularly the astrocytes, the system's function. So basically, it is a macroscopic, glial-dependent waste clearance system that utilizes the unique system of perivascular channels formed by astroglial cells to promote the efficient elimination of soluble proteins and metabolites from the central nervous system. Fascinatingly, the glymphatic system functions mainly during sleep and is largely disengaged during wakefulness. The biological need for the sleep across all species may therefore reflect that the brain must enter the state of activity that enables elimination of potentially neurotoxic waste products, including beta amyloid. Since the concept of glymphatic is relatively new, we will here review the basic structural elements, organization, regulations, and the functions. Function of glymphatic system. Waste clearance. The primary function of glymphatic system is to clear waste products from the brain. This includes the removal of metabolic waste products, protein, and other debris that accumulate the brain's interstitial fluid. One of the most well-known waste products removed by the glymphatic system is beta amyloid, which is associated with Alzheimer's disease. Fluid regulation. The glymphatic system helps to regulate the flow of cerebrospinal fluid, that is CSF, through the brain tissue. It facilitates the exchange of CSF with interstitial fluid, allowing the removal of waste and delivery of nutrients and other substances to the brain cells. Thirdly, sleep-dependent process. The glymphatic system is most active during sleep, particularly during slow wave sleep. This suggests that it is an essential component of the brain's nightly maintenance and waste clearance mechanism. So, we might think, other than the elimination, this doesn't have any other function. No, we are wrong. Besides waste elimination, there is another function, that is, glymphatic system may also function to distribute non-waste compounds such as glucose, lipids, amino acids, and neurotransmitters related to volume transmission in the brain. Coming on to the regulation of glymphatic system. The glymphatic system is regulated by various mechanisms and factors including astrocytes. Glial cells called astrocytes play a crucial role in the glymphatic system. They have specialized water channels known as aquaporin 4, channels that facilitates the movement of CSF through the brain tissues. Pulsation. Cerebral blood vessels pulsate, and this pulsation helps drive the movement of CSF through the brain tissues. This pulsation, in part, facilitates waste clearance by the glymphatic system. Sleep. As mentioned earlier, the glymphatic system is most active during sleep, particularly the slow wave sleep. The expansion of interstitial space during sleep allows for more efficient waste removal. Blood-brain barrier. The blood-brain barrier regulates the entry of substance into the brain. The glymphatic system allows the exchange of certain substances between CSF and the brain tissue, aiding in the waste clearance. Lastly, aging and disease. The glymphatic system's function may be compromised by aging and in various neurological conditions, such as Alzheimer's disease. This can result in the accumulation of toxic substance and contribute to the disease progression. As you can see the image here, it's a schematic representation of brain's fluid compartments and barriers. The fluid compartments in the brain consist of intracellular fluid 60 to 68%, interstitial fluid or extracellular fluid 12 to 20%, blood 10%, and cerebrospinal fluid 10%. The blood is separated from CSF and interstitial fluid by the blood-brain barrier and blood-CSF barrier, respectively. Tight junctions between blood endothelial cells constitute the blood-brain barrier, restricting the macromolecules to move freely from the blood to the brain parenchyma. Fluids and solutes in the perivascular space located between endothelial cells and astrocytic end expressing the water channels aquaporin 4 diffuses into the brain parenchyma. The blood-CSF barrier is formed by the tight junctions between the choroid plexus epithelial cells. Macromolecules from the blood can move freely between fenestrate endothelial cells to interstitial fluid, but it is restricted by the tight junctions in the choroid plexus epithelial cells, which therefore are believed to the main players in determining the CSF composition. Let's see how the lymphatic system relates to CSF and the removal of waste products. Cerebrospinal fluid circulation. The CSF is continuously produced within the brain's ventricles by the choroid plexus, as previously explained. It then circulates through the ventricles and subarachnoid spaces surrounding the brain and spinal cord. CSF entry to the brain tissues. CSF flows the brain tissue and fills the space around the blood vessels and neurons. This provides essential nutrients and removes waste products from brain cells. Interstitial fluid exchange. The glymphatic system involves in the exchange of CSF with interstitial fluid. The interstitial fluid is a fluid that surrounds a neuron and other brain cells. The exchange of these fluids occur primarily in the perivascular spaces, which are narrow channels around the blood vessels in the brain. Active transport and aquaporin 4 channels. The exchange of CSF and ISF is facilitated by specialized tissues called astrocytes. These cells have aquaporin 4 water channels that allows the regulated flow of fluids between blood vessels, CSF and the brain tissue. Astrocytes actively transport specific substances such as iron, water to regulate this exchange. Waste clearance, as above mentioned. The glymphatic system is more active during sleep, particularly during slow wave sleep. During this time, the interstitial space in the brain expands, allowing the more efficient clearance of the waste products. This includes the removal of metabolic waste, soluble proteins, and other substances that can accumulate in the ISF. Transport of waste products. Waste products are removed from the interstitial fluid, are transported into CSF. These waste products are then carried away through the circulation of CSF and eventually reaches the bloodstream for elimination from the body. Regulation and impact. The glymphatic system is influenced by various factors such as the activity of astrocytes, arterial and venous pulsation, and overall sleep-wake cycle. Disruption of glymphatic functions can have implications for accumulation of toxic substances and development of neurological conditions such as Alzheimer's disease.
But how does failure of the lymphatic system cause neurodegenerative diseases and other problems to the brain? To begin talking about how it can affect the brain, we'll start talking about a simple neurodegenerative disease called Alzheimer's. Alzheimer's is one of the most common types of dementia. It is a progressive disease beginning with mild memory loss and possibly leading to the loss of the ability to carry on a conversation and respond to the environment. Causes of Alzheimer's disease include toxic waste due to an accumulation of toxic wastes within our brain, which is usually manifested in old age. These toxic wastes can be apolipoproteins, beta amyloids, tau proteins, etc. So what does our body do to remove all these harmful beta amyloids and other toxic wastes? The glymphatic system is a recently discovered macroscopic waste clearance system that utilizes a unique system of perivascular channels formed by astroglial cells to promote efficient elimination of soluble proteins and metabolites from the central nervous system. Our body, through the power of evolution and lots of trial and error, went on to create the glial lymphatic system, or aka the glymphatic system. It is a highly polarized CSF and interstitial fluid transport system that is mainly active when we sleep and facilitates extracellular waste removal through a network of astroglia supported perivascular or perineural channels that drain into the lymphatic networks of the brain or the major dural sinuses. It drains away all the neurotoxic waste our body produces in our brain, for example apolipoproteins, beta amyloids, etc., which then damages the nerve cells due to excess accumulation. So basically, the glymphatic system is the brain's drainage system. Now, why do some people with trauma get Alzheimer's and other kinds of neurodegenerative diseases? Traumatic brain injury induces a release of beta amyloid peptide and CTOR, a proteolytically cleaved product of MAPTOR, which is a highly abundant intracellular microtubule protein in axons. CTOR is a biomarker of brain injury since it is released in vast quantities and correlates with severity of traumatic brain injury. A hypothesis says that because of the high quantities of TOR, it could lead to more cellular uptake and cause it to start making fibrillary aggregates, which then attracts and compounds itself to more TOR, which would then become neurofibrillary tangles, which would cause a prion-like pathology. Traumatic brain injury is also linked to formation of large astroglial scars and persistent activation of innate neuroinflammation, which could also lead to brain damage. Now, how can the glymphatic system take care of that? The glymphatic system is extremely useful in the regenerative phases after an ischemic stroke by eliminating all the deposition and hyperphosphorylation of TOR. This prevents post-stroke dementia. This also promises the use of GS of glymphatic system for therapeutic purposes, which can then be perfected in the future. The brain, to prevent any damage to occur in it, inactivates certain regions of the brain and induce a comatose state so as to conserve energy, just like how cardiac cells hibernate when the heart suffers a stroke. Interestingly, a murine study evaluating clearance of fluorescent traces from the necrotic infarct core at 7 weeks after hypoxic middle cerebral artery occlusion showed that solutes trapped in the core leak through the glial scar spaces are transported from brain along the perivascular spaces.
So at this related point of time, we get the main role of glymphatic system. What does it do? How does it function? How does it produce? But what if there's a functional error? It's the most common form of dementias existing, and most foremost is Alzheimer's disease, and the second one goes for Parkinson's. But there are reasons for it. For example, accumulation of misfolded proteins will keep on seeing over the presentation. But if there is any particular functional error in the glymphatic system, which is the proper drainage system for our brain, which collects all these wastage and goes and recycles it, what happens if it's a functional error? First of all, what is Parkinson's disease? Parkinson's disease is a basic demential condition where happens due to substantia nigra degradation. What is substantia nigra? Substantia nigra is basically a part of basal ganglia which produces a bunch of parts of brain which produces dopamine. So what happens when it degrades? When it degrades, the production of dopamine reduces. Why is so important? Because dopamine is responsible for movement, memory, and pleasurable reward and motivation. And when we come for memory and movement, that's where most of the indications of Parkinson's resides. For example, people with Parkinson suffer for uncontrollable movements such as shaking, stiffness in the muscle, difficulty with the balance, coordination, confusions as well. So how does this Parkinson occur? It can happen not only 100% genetics but due to environmental effects as well, such as stress or exposure to any particular toxins. So when does it start? Usually it starts around at the age of 60, and it is due to lack of the autophagosums and other microglial cells that's not properly doing its work. But there are exceptional cases such as known as juvenile Parkinson's disease. So this happens under age of 21. So that is the basic concept of Parkinson's. So now let's put the shades of perception under the aspects of glymphatic system. What is this? How does it primarily contribute to this Parkinson's? So there are different ways that Parkinson's could develop, but primarily it's due to protein misfolding, as I've said before, such as beta amyloid, alpha synuclein, tau proteins. And regardless of protein misfolding, there's a dysfunction of mitochondria as well. So mitochondria is a dynamic cell organelle. It could join together, separate together to do a particular task. So impairment such mitochondrial function can increase oxidative stress, which can exactly go towards the protein modifications. And now we're going to take a closer look on the protein misfolding. So under Parkinson's, we are going to talk about Lewy bodies. What are Lewy bodies? Lewy bodies are aggregations of these misfolded proteins that is present in the cell bodies. So where does it come from? So it's a precursor that is forming from dystrophic neurites. And what does it consist of? It consists of mainly two stuff, that is ubiquitin and alpha synuclein. And now we're going to talk about mostly alpha synuclein because that is one of the formation and aggregation that is particularly present in the Parkinson cases. So what are these alpha synucleins? Alpha synucleins are basically these neuronal proteins that regulate synaptic vesicle trafficking and as well neurotransmitter release. So at a point when it's under the limit, it is very good for the body. But what happens when there is genetic variation, or what happens there is a particular misfolding happens? So at that time, it aggregates as a oligomers and it finally forms fibrils. So what happens in these fibrils? These fibrils aggregate together and dysfunction the particular neuron. And if neuron goes under dysfunction, it basically dies. This misfolded alpha synucleins, they do not present in the neuron which is dead. It spreads through the neurotransmitter to other neurons. And basically what it does, it blocks it again, and there's a lot of blockade happens and the neuron dies, and essentially it spreads. And moreover, what happens is, when such processes releases such misfolded proteins, sometimes it goes bind with microglia and astrocytes present in the brain parts. So what happens then? These releases the particular thing called cytokines. These cytokines essentially creates inflammations in the neurons and make it much more worse and essentially leading to the death of neuron. So these are the particular theories that there could be leading to Parkinson's. Till now, we do not have any particular treatment to stop it, but there are treatments to reduce the pace of it. And one more last point I want to mention is the part aquaporin 4. So aquaporins, what are these aquaporins? These aquaporins are basically protein channels which are present in all over the body, and there are around 10 aquaporin channels. But in central nervous system, mainly aquaporin channel 4 is present, and it is mainly responsible for letting in cerebrospinal fluid into the parts of brain, where it mixes with the interstitial fluid and clears out the debris or misfolded proteins or any kind of wastage that is present over there. If any dysfunction happens in this aquaporin 4 channel, it could lead to the dysfunction of the entire glymphatic system. So these are the ways that could affect the particular neurons that could lead to such demential conditions. And as I mentioned earlier, there are treatments for it to pace it down, but there isn't any treatment for it to stop, because till now we do not know the particular way that's how it's produced or how it is prolonged such way that demential conditions